I'm going to be presenting the following webinar on T-cell characterization, combining advanced flow cytometry with live cell imaging. I will start by giving an overview of advanced flow cytometry and how it is being used today in biological applications, then touch on important stages of T-cell biology and how that relates to the immuno-oncology therapeutic area, as well as how those processes could be evaluated. Finally, I will discuss two workflows, one of which will focus solely on advanced flow cytometry, and the second will suggest a method of how flow and live cell imaging could be combined to provide deeper insight into T-cell biology and how it can be modulated by therapeutic drugs. Advanced flow cytometry has evolved into a technique for rapid analysis of DNA content, cellular marker expression, as well as electronic sorting of cells of interest for further investigations. It has been extensively used for monitoring phenotype expression, drug transport, cell cycle, proliferation, and apoptosis, to name a few. It has also become an important tool for both diagnostic and therapeutic monitoring of patients. The importance and contributions of advanced flow cytometry in the drug discovery world, particularly immuno-oncology, is only becoming greater. The iQue advanced flow cytometry platform allows the user to streamline research workflows and achieve fast, actionable results from samples. This is enabled by our advancement in the following key areas: speed, content, miniaturization, usability, and insight. The iQue system itself has a patented sampling system which allows for rapid throughput and plate processing, and iQue assays have been designed to be mix and read for ease of use. Incucyte software is intuitive and provides a single interface from assay design to analysis. It also allows for fast analysis, and when assay kit templates are used, the data is pulled through automatically so users can focus on assay design. The kits designed for use on the iQue allow the user to perform miniaturized multiplex assays, as well as perform both phenotype and function quantification and the analysis of secreted proteins. All of these attributes combined give greater insight into the biology being studied and ultimately create the environment for better decision-making. Sartorius offers solutions to many different scientific research areas, including antibody discovery, foundational approaches such as cell health, and immuno-oncology. Immuno-oncology is the study and development of treatments that utilize the power of the body's immune system to fight cancer. This relatively new therapeutic area has grown immensely and has given rise to some of the most exciting drugs of the last 20 years, including CAR T-cells. Advanced flow cytometry has been at the center of these discoveries, not only because of the rich data that can be achieved through screening, but also the ability to analyze and convert data into meaningful endpoints, i.e. drugs that work. The T-cell subpopulation has probably been the most heavily focused on so far, and therapeutic drugs such as monoclonal antibodies that include both BiTEs and checkpoint inhibitors have been developed, which activate or enhance activation of this subset of immune cells and invoke their seek and destroy instincts to target cancer cells. To further develop these kinds of drugs, there is a need to be able to fully assess how they work in terms of characterizing their effects on T cells in physiologically relevant assay models. Assaying on the iQue allows for the analysis of multiple cell types and provides readouts including T-cell phenotype and secreted proteins such as cytokines. In order to fully explain how these capabilities help in T-cell characterization and ultimately better drug design, I will now give a brief summary of the important stages of T-cell biology, which are the main focus of the therapeutic drug discovery process. A naive T-cell is a T-cell that is differentiated in the bone marrow, having successfully undergone countless positive and negative processes, central selection in the thymus. Among these are the native forms of helper CD4 positive and cytotoxic CD8 positive T cells, and they have not yet encountered a cognate antigen. Activation occurs through the recognition of an antigen on the surface of antigen-presenting cells, or APCs. The T-cell receptor, or TCR, found on both CD4 and CD8 positive T cells binds to the antigen, which is held in place by a structure on the APC known as the major histocompatibility complex, MHC. This triggers the initial activation of the T cell and causes increased expression of certain surface markers, such as CD69, 25, and HLA-DR, as well as the release of cytokines such as interferon gamma, TNF-alpha, and also interleukin-2. Once activated, CD8 positive cytotoxic T cells then respond by moving to the site of infection or inflammation to destroy the pathogen, for example, an infected cell. The CD8 positive T cell recognizes the pathogen by the foreign antigen that is presented by the class 1 MHC molecule. The TCR and CD8 receptors then bind to the MHC, which then allows the T cell to create a pore in the infected cell's membrane and inject granzyme B, known as degranulation, which induces apoptosis and the infected cell dies. T cells are still expressing activation markers here, as well as CD107A, which indicates degranulation, as well as FAS ligand, which is also required for induction of apoptosis. Sometimes a cytotoxic T cell can enter into a state called exhaustion. This is a broad term used to describe the response to chronic antigen stimulation. It is a dysfunction defined by poor cytotoxic function and is characterized by a decrease in cytokine release, namely interferon gamma and TNF-alpha, expression of exhaustion markers such as PD1, TIM3, and LAG3, as well as low proliferative properties. The tumor microenvironment has been found to promote T cell exhaustion in T cells, and ideally this state could be reversed so that the cells could regain their cytotoxic capabilities, and studies into checkpoint inhibitors have shown this is possible. Once the pathogenic response has been neutralized, the majority of T cells will then quickly die off, leaving behind a small pool of memory cells with heterogeneous phenotypes and functions. Reinfection with an antigen that is recognized by these memory cells then allows for rapid expansion of specific cytotoxic T cells that will seek and destroy the antigen before an infection develops. It is these key areas that are being focused on for the development of drugs which aim to modulate the biology. Sartorius have developed 4 T cell and cytokine profiling kits so that these areas can be investigated using advanced flow cytometry. Each kit has a similar assay concept and are pre-optimized to reduce experimental setup time. They are robust mix and read assays and use miniaturized volumes to reduce precious sample use and have a pre-designed gating strategy, including compensation for ease of use, and are available as both 96-well and 384-well assays. These are single-well assays which give the following readouts of viability, proportions of T cell subtypes, T cell status markers, and cytokine secretion. The kits include an antibody cocktail mix and viability dye, which are added to samples for labeling. The first of these 4 kits is the T cell activation cell and cytokine profiling kit. As you can see in the diagram to the right, both CD8 and CD4 T cells can both be identified, as well as their expression of early, late, and later activation markers CD69, 25, and HLA-DR, respectively. Q beads allow for the determination of interferon gamma and TNF-alpha secreted cytokine concentrations. The T-cell-mediated killing kit facilitates a co-culture model where an encoding dye is included so that target cells can be identified and their death through interaction with activated T cells can be quantified to give a functional readout. CD25 positive CD8 cytotoxic T cells are able to be identified, as well as granzyme B and interferon gamma release. T cell exhaustion allows for the identification of the exhaustion markers PD1, TIM3, and LAG3 on both CD8 and CD4 T cells, and also looks at both interferon gamma and TNF-alpha release. Finally, the T cell memory kit is able to identify up to 7 different memory cell phenotypes, as well as interferon gamma and interleukin-10 release. The described kits can be mixed and matched in order to create a bespoke T-cell characterization panel to suit the user's particular needs. This is particularly amenable to testing iotherapeutics, which focus on inhibiting or enhancing effects in these particular areas of T-cell biology. Because these assays have been miniaturized, a single assay can feed up to all 4 of the kits if necessary. This next slide uses the T-cell activation kit as an example of how the data is handled by Foresight. Firstly, the Q beads and the cells are separated. Again, these gates are preloaded into the software, so apart from slight adjustments to their location, no other activation is needed to generate this strategy. We can also separate labeled target cells from PBMCs using the BL1 channel. It should be mentioned here that this separation can be performed for all the kits if necessary, which is what we have done in the following workflow. The next gate shows viability gating. Both antibodies and viability stain concentrations have already been optimized, so fluorescent signals will already be in range. Next, we start to identify the T cells, then gate into their subtypes, and finally their activation status. As mentioned earlier, all of these gates are already in place. However, inclusion of positive and negative control wells allows for users to check the gate is in the best possible position. I will now describe a workflow that details how we have used these assays to characterize the effects of activators on T cells. Co-culture of PBMCs and suspension target cells, which were nut-like green labeled Ramos cells, were plated in a 96-well plate. Cells were then treated over a 72-hour period with increasing concentrations of either CD3, CD28 dynabeads, or staphylococcal enterotoxin B. 10 microliter samples of cells and supernatant were taken from the assay plate and transferred into 4 96-well V-bottom plates. Each T-cell characterization kit was added to one of the 4 sample plates. Data from each labeled sample plate was acquired using the iQue. Each kit comes with its own specific template to be loaded up onto Foresight so that the data can be pulled through automatically. By using the described gating strategy in the earlier slide, we can see a summary of the kind of data that can be produced firstly from the T-cell activation kit. The 2 graphs on the right show the activation markers CD69, 25, and HLA-DR as a percentage of CD8 positive T cells. The graph to the left represents dynabead activation, where beads were added as a ratio to effector cells, with the top bead concentration representing a 1-to-1 bead to effector cell ratio. The graph to the right shows SAB concentrations in nanograms per mil. Clear differences can be seen in the T-cell activation profile, where dynabeads show a concentration-dependent increase in all 3 activation markers, whereas SEB activation presents a much flatter activation curve. Cytokines can also be used to monitor T-cell activation, and analysis from the kit here shows both interferon gamma and TNF-alpha release in response to the activators. It is also possible to add extra Q beads to these assays by adding Q bead plex screen kits, which allow for multiple cytokines to be analysed simultaneously. Again, even very low concentrations of SEB produce extremely high concentrations of both cytokines, whereas dynabeads induced a concentration-dependent increase in cytokine release. SEB is a superantigen and will elicit an extreme response in terms of cytokine release. However, being able to predict these kinds of responses in immuno-oncology drugs is very important in order to reduce adverse side effects such as cytokine storms, which can be fatal. As mentioned earlier, the killing kit gating strategy allows for the separation of green labeled target cells from the immune population and calculation of target cell viability, which has been plotted up here. As expected, the dynabeads induced a concentration-dependent decrease in target cell viability, whereas SEB activation resulted in similar levels of killing across the different concentrations. This difference was again due to SEB potency, creating blanket high cell death over all concentrations and low cell target numbers, creating this flatter effect. Cytokine analysis can again be assessed using the killing kit. Here, Foresight allows for the generation of heat maps to quickly and effectively give the user an overview of the biology occurring. Granzyme B release confirms the likelihood that the cell death occurring in this assay is due to T cell degranulation, as does the increase in interferon gamma. This data is then able to be easily expressed to compare the activators, and as part of Foresight's template, CRCs can also be generated, giving important pharmacological results to aid in making important decisions quickly. As well as developing drugs to enhance activation of T cells, being able to prevent exhaustion is also an area of interest, and checkpoint inhibitor studies using monoclonal antibodies are proving promising. The T-cell exhaustion kit allows for the evaluation of this phenomena by being able to detect changes in key exhaustion markers PD1, LAG3, and TIM3 on both CD8 and CD4 T cells, as well as the cytokines interferon gamma and TNF-alpha. Foresight software again allows for various presentations of the data, including being able to compare individual gated populations in a plate view, which allows for the opportunity to spot outliers and check that controls are behaving as they should. The final area that this workflow covers in terms of T-cell characterization is the T-cell memory development. This kit allows for the identification of the various subtypes of T memory cells that develop as a result of stimulation. The image on the bottom right shows how this progression drives T cells ultimately towards a more differentiated phenotype, which has decreased self-renewal properties. The long-term success of IO therapy, such as adoptive transfer of T cells, is ultimately dependent on the ability to produce highly differentiated memory T cell subsets, which will persist in their presence and thus be able to generate a response to any re-emerging cancer cells. Being able to know whether this will happen in vitro is an important tool to have in order to predict what is likely to happen in patients. The analysis shown here details how our two activators have affected the T-cell memory populations. Both Dynabeads and SEB stimulation shows how T memory cell development has been pushed towards a more terminally differentiated phenotype. As stimulation increases, the naive population is rapidly diminished as expected, as are the more stem cell-like cells. In turn, there is a concentration-dependent increase in the more terminally differentiated populations. IL-10 cytokine is heavily involved in T-cell memory immunoregulation, and its release is also increased in response to stimulation. This case study was an example of how the iQue can be used to characterize T-cell as a standalone instrument. What I'm now going on to describe is how it can be used in combination with the Incucyte Live-Cell Analysis system. How the two instruments work together is highlighted here in this diagram, which shows the positioning of each in assay development and drug discovery. iQue's strengths of being able to rapidly sample multiplex high-content assays allow for lead profiling and identification, which can then be complemented by Incucyte's ability to then perform further lead characterization using kinetic measurements, and at the front end of assay development, where the kinetic analysis can be used to pinpoint the best time to sample and analyze with the iQue. Just to go into some detail as to how the Incucyte Live-Cell Analysis system works, we can see here to the right that it consists of a main gantry which sits inside the incubator to provide the optimum environment for uninterrupted cell growth. The gantry houses a shelf for holding cell culture vessels, as well as the objectives to allow for images consisting of phase contrast and up to 3 fluorescent channels to be taken at intervals decided by the user. The control panel is responsible for data analysis and sits on top of the incubator. Incucyte software is then able to connect to the controller so that users can then set up experiments and perform analysis at the desk. So now that we've had a brief introduction to the Incucyte, I will now describe how the iQue and Incucyte were used to interrogate an immuno-oncology therapeutic antibody known as a BiTE and show how it modulates T-cell activity. BiTE stands for bispecific T-cell engager antibodies, and they are a class of artificial bispecific monoclonal antibodies that are used as anti-cancer drugs. They work by bringing T cells and target cells together by forming a link between the two cell types. The BiTE are fusion proteins consisting of 2 single-chain variable fragments of different antibodies, shown in pink and yellow. The particular BiTE used in this study takes these fragments from an antibody CD3, shown in pink, which allows for T-cell binding, and is generally the surface marker used to construct these antibodies, and then uses CD19, shown in yellow, which is an antigen commonly found on liquid tumours such as non-Hodgkin's lymphoma. The therapeutic version of this drug is called blinatumomab. However, we are using a biosimilar here. Upon binding to the target and effector cell, the T cell then becomes activated and causes the release of cytotoxic molecules into the target cell, causing its death. This directed form of T cell activation is MHC independent. This is an important modification as tumour cells evade the immune system by down-regulating MHC antigen expression, which is what T cells normally require for activation. The T cells are able to proliferate upon this activation with the BiTE in order to effectively kill the target cells. In order to study the effect of this antibody on target and effector cells using both instruments, we firstly developed and optimized a workflow that allowed their connection so that the same cells could be analysed from a single assay plate, firstly by the Incucyte, and then at an appropriate time the iQue. The green labelled target cells are firstly seeded into a 96-well plate. Activators and effector cells are added next, and if desired, an Nectin-5 red in order to monitor apoptosis. Once the experiment is set up, it can then be placed into the Incucyte for imaging. During imaging, 10 microliter samples can be taken from the supernatant in order to collect temporal data on cytokine release. Because this sample size is so small, a number of samples can be taken without affecting the assay itself. At an appropriate time point, the assay can be removed from the Incucyte for analysis on the iQue. The plate is centrifuged and cells are washed with PBS. Accutase is used to gently lift the cells from the plate, and the cells are then transferred to a V-bottom plate, where they can be labelled with antibodies. In this particular case, the TCA kit was used to characterise the T cells. So temporal cell health, morphological and spatial quantification can be gained through the first part of the workflow with the Incucyte and detailed T-cell interrogation from the iQue. Here, nut-like green-labelled Ramos cells were co-cultured with non-labelled PBMCs. The co-culture was treated with increasing concentrations of BiTE antibody for 72 hours. The images shown here are representative of the co-culture in the presence of 10 nanograms per ml of the BiTE antibody at 0 and 72 hours. Incucyte cell-by-cell masking, shown here in yellow, can be used to achieve an individual object or cell count. The size and shape of these masked objects can also be quantified, as well as fluorescence intensity and area. As the PBMCs were not labelled and the Ramos are green, the masking is capable of distinguishing between the two cell types. It is then possible to quantify phenotypic changes in each population in terms of cell health, as well as spatial and morphological data. In these two images, we can see how the Ramos cells start as individual green objects, but are then rounded up into clusters by the activated PBMCs. This change in behaviour can then be quantified by looking at the fold change in green integrated intensity, and this metric allows for the fact that we lose definition of the individual green objects as they cluster, and when this data is plotted over time, we can see that the BiTE antibody induces a time-dependent, concentration-dependent decrease in green signal, meaning the target cells are dying in response to the presence of the BiTE. This data can then be used to achieve pharmacological readouts, and we see here that only the BiTE-induced death and the control antibody used here, which was a CD3 beta-galactase BiTE, did not induce such a response. BiTE activation can also be visualized by looking specifically at morphological changes. BiTE stimulation was compared to the well-characterized diabead activator beads. These images of the co-culture were taken at 72 hours and compare the two activators to the control antibody mentioned earlier. In the last slide, the green intensity was used as an indicator. However, here, average cluster size can be used. The arrows show how the BiTE and dynabeads induced larger clusters when compared to the control, which can again be quantified and also plotted against time if desired. The activated PBMCs also increased in size, which can be seen clearly in these images and quantified. This is also corroborated by the data taken from the same 72-hour time point and analysed using the iQue. A forward scatter histogram showing the three populations validate the observation from the Incucyte, with the two activators generating very similar responses, where we see a shoulder appear that represents the larger subpopulation of the activated T cells. Side scatter histograms also show the change in granularity was highly comparable between the activators. Daily cytokine release from the T cells was also compared using the iQue. The graphs on the right show both activators induced a time and concentration-dependent increase in both interferon gamma and TNF-alpha. However, the top bead concentration included in the graphs here, depicted as the stripe bar, caused an early release of these cytokines when compared to the BiTE antibody, as well as much greater concentrations. The antibody concentrations are not the same, so therefore these results cannot be directly compared. However, when the last time point from the Incucyte data is taken into account, we see that although there is much greater cytokine release through dynabead activation, the BiTE antibody actually achieves a greater level of killing. T-cell activation markers were also assayed, which show both activators were capable of producing a response. Spike antibody appeared to produce better defined activation curves for all three markers when compared to Dynabeads, potentially suggesting that at the 72-hour time point, T-cell activation may have advanced faster with the BiTE antibody. This theory is supported by the kinetic data from the Incucyte, showing that killing with the BiTE also occurred at a faster rate when compared to the Dynabeads. The light grey time course shows the BiTE point of inflection at approximately 50 hours, whereas the Dynabead shown in the dark grey only begins to show a decrease in target cells at the last time point, again suggesting that the T-cell activation has progressed at a faster rate. The data supports that BiTE activation not only effectively activates T-cells, it can kill more effectively and faster than the more crude activation from Dynabeads, without producing excessive amounts of cytokine. When designing therapeutic drugs, being able to detect for such attributes is an important ability, and having the tools to carry this out effectively even more so. In conclusion, the workflows described here show how advanced flow cytometry can be used to effectively characterize and profile T-cells, and coupling this capability with reagents that are easy to use and software that allows quick on-the-fly decision-making can drastically speed up the drug discovery process. Further development of these workflows involves the marriage between different technologies that not only complement each other but also validate results. Using both advanced flow cytometry and live-cell analysis allows to better characterize T-cell subpopulations, which in turn creates better IO drugs faster. There are links here for further information on this area, and thank you for listening.